The overall goal of the following experiment is to investigate the substrate specificity of protein lysine methyltransferases, or PKMTs. This is achieved by identification of an initial substrate and preparation of peptide arrays with several variants of this substrate containing single amino acid exchanges at different positions. As a second step, the peptide array is incubated with the PKMT in the presence of a radioactive methyl group donor, which allows detection of peptide methylation by transfer of radioactivity to the corresponding peptide spot. Next, the array is exposed to an X-ray film and quantitatively analyzed in order to determine the methylation activity at each of the peptides and to define the specificity profile of the PKMT. The results show which amino acids in the substrate peptide are critical for its methylation by the PKMT.
The main advantage of this technique over existing methods like methylation of individual peptides is that a number of different peptides can be tested in one experiment in a cost-efficient manner.
This method can help answer key questions in the protein lysine methylation field, such as the identification of previously unknown novel substrates of PKMTs.
Though this method can provide insight into the specificity of PKMTs, it can also be applied to other systems such as protein kinases, other protein methyltransferases or acetyltransferases.
I first had the idea for this method when I became familiar with the possibility of preparing large custom peptide arrays at reasonable cost using peptide spot synthesis.
To begin this procedure,